enzymes. Okay, so enzymes, are they protein? Yes, they are protein. But in this session, I'm not going to cover into much detail, just going to give an overview of enzymes and their importance in the food system. Enzymes play an important role in our food systems. Normally, it's an indication of food quality. When we talk about enzymes in food, it can be beneficial or it can be detrimental to the quality of our food. Okay, most of the spoilage, for example, milk spoilage and whatnot, is due to the enzymatic activity of the endogenous enzyme that you can find in milk. So this will rapidly reduce the shelf life of the food. But in some cases, you want to utilize enzymes in order for you to biotransform, okay, food component into another favorable compounds, okay, or you want to use enzymes to hydrolyze, to break down starch into reducing sugars. Okay, we also can use enzyme as a processing tool and utilize it in the waste management as well. Okay, not only that, on the analytical part, we can measure the activity of enzyme, okay, by utilizing enzyme as part of the analytical methods, or we can quantify food constituents, okay. For example, good example for this will be ELISA, where you conjugate this enzyme to an antibody in the presence of specific proteins or analyte. This antibody will bind to this analyte and allow this enzyme, okay, to be bound as well, and when you add substrate, it can turn into a colored product. So this is one of the function of enzyme that we can use. Or we can also use enzymes to quantify inhibitors. Okay, so it is in food science, in food system, it's important to isolate and characterize enzymes and for us to know what are the enzyme kinetics. Okay, so by definition, enzyme is a biological catalyst that is capable of specific activation and conversion of substrate to product. So here we have substrates that is going to be hydrolyzed by enzyme to produce product, and this reusable enzyme will then repeat the process for the next substrate. Okay, so one thing we need to know about enzyme is that they are proteins. So all the components of proteins, all the physical chemical properties of proteins is going to be inherited by an enzyme, meaning that you can denature protein, it can lose its activity if the enzyme being denatured due to temperature or pH and so on and so forth. Okay, so the way we measure enzyme, okay, we measure them as standard unit. We call this as U unit or international unit IU. So 1 IU of enzyme catalyzes the transformation of 1 mole of substrate to produce per minute under standard condition. Okay, enzyme may stand alone as a protein, or in some cases it has to be complexed with coenzyme, okay, for it to become a whole active enzyme. So the protein portion of these types of enzyme, we call it as apoenzyme. Okay, so enzyme in this form is still inactive. Okay, and then we have cofactor, which is a coenzyme. This is normally the non-protein portion, and they act as activator of the enzyme. And by having this complex of the coenzyme and apoenzyme will give you this holoenzyme. Okay, so holoenzyme is an active form of the enzyme that can then be used to catalyze reaction with the substrate. Okay, the activity of enzyme is highly specific. So the enzyme must bind to the substrate stereoscopically and correctly into the active site before any catalysis can occur. So there are two types of theory behind this enzyme specificity. It could be lock and key theory by Fischer. So in this theory, enzymes are specifically complex and they have this very rigid active site. Okay, so the rate of this enzymatic reaction is affected by the removal or small change in configuration of a group. So if it's not a good fit, this enzymatic process is not going to occur. There's another theory on enzyme specificity would be by Koshland 1959 called induced fit theory. Here, the enzyme is stereospecifically complex, but different than the lock and key theory is that the active site here of the enzyme is not rigid. It is flexible to accommodate between enzyme and substrate when they form complex. So if you look at this figure down here, okay, it's just a summary of how this induced fit theory of enzyme work. So first you have substrate that enter the active site of enzyme, okay, to form this enzyme substrate complex. So here, for the induced fit theory, the enzyme will change its shape slightly to allow the binding of the substrate. So now this hydrolysis will occur, so enzyme will become enzyme products complex, and the products will leave the active site of the enzyme. So there are also regulations on the enzyme reactivity. We can look at the velocity of enzyme reactions, which is directly proportional to the enzyme concentration. And the velocity of enzyme reactions is dependent on the concentration of the substrate, the inhibitors, as well as cofactor. And it can be affected by change of pH. Okay, the velocity of the enzymatic rate reactions can be reduced by 0.5 times if you change the pH unit by 1 from the optimum pH. And temperature can also reduce the velocity by 1.4 to 2 times as you reduce the temperature by 10 degrees Celsius. It is important for you to understand the enzyme kinetics in order to control rate of enzyme catalyst reaction. So enzyme activity can be further explained by this Macaulay-Menten kinetics, where it involves at least a minimum of 2 events, where the enzyme and substrate will be stereochemically bind, and this relates to the chemical conversion. So the binding of the substrate to enzyme is assumed to represent equilibrium conditions between the associations of enzyme plus S will lead to this enzyme substrate complex, and the dissociation step, okay, from this, let me just show you here, from the enzyme substrate complex to the enzyme plus substrate, each with the respective and characteristic second order, okay, which is K1, and the first order K minus 1 rate constant. So we can actually calculate the rate of enzymatic reaction. For example, we can determine what are the initial velocity. So what is initial velocity? This can be obtained as the slope of the curve, okay, following the pre-steady state period in the enzyme reaction. Okay, and we also need to determine what are the maximum velocity or Vmax. So this is the maximum velocity. So maximum velocity can be affected by the nature of substrate, okay, your enzyme concentration, changes in pH, temperature, and also the presence of certain types of inhibitors. Okay, this is going to affect the maximum velocity. Okay, so in enzymatic reactions, we can also measure the Michaelis constants, or Km. So what is Km? Km is the concentration of substrate at half of the maximum velocity. Okay, so the unit for this Km, or Michaelis constant, is in concentration, okay, milligram per mil, or it could be milligram or gram per liter. It really depends. Okay, so enzyme that has higher Michaelis constant will have lower affinity for its substrate, and what it means is that it will need a greater concentration of substrate to achieve Vmax. So here I show you an example of these two enzymes. Okay, you have enzyme 1 in this brown line and enzyme 2 in this green line here. Okay, so if you see here, enzyme 1 has lower Michaelis constant value compared to the enzyme 2. Okay, so which one do you think would be better, okay, and has higher affinity? Right, the answer will be enzyme number 1. Okay, enzyme 1, because it has lower Michaelis constant value, so means that it requires less substrate, okay, for it to achieve this maximum velocity. So in food science, it really depends whether having low Michaelis constant value is good or not. Okay, so the good consequences is that if you desire that enzyme reaction, for example, you want to produce dextrin, you want to in the production of dextrose equivalent corn syrup using amylase, okay, conversion from starch into this simple sugar. So you want your enzyme to have low Michaelis Menten, because means that this enzyme has higher binding affinity, and the rate that reach half of the maximum velocity will be at a lower substrate concentration. This will help in increasing the productivity of the enzymatic reaction. What it means here is that you will produce more of your product, which is a simple sugar, in shorter time as the velocity is larger. So having a small Michaelis constant means higher binding rate, as enzyme is saturated at low concentration of the substrate. But for those undesirable enzymatic reaction, for example, the enzymatic browning of fruits such as banana, okay, so if you have low Michaelis constant value, okay, for the substrate, means that you have higher binding affinity, means that low substrate level can reach maximum rate. So what you can do to slow down this enzymatic reaction is by adding inhibitor. Okay, so here the velocity is high at low concentration of substrates. Okay, so for two enzymes that react on the same substrate to produce the same desirable product, you need to choose one that have smaller Michaelis constant in order for you to have more yield of your product per hour. Unless in your case your substrate concentration is high, then you can choose either one of these enzymes. Okay, so the rate of enzymatic reactions, from here you can actually use to measure the amount of substrate needed or to measure the activity of the enzyme. For example, when the concentration of substrate is less than the Michaelis constant, this means that the rate of reaction is first order with respect to the substrate concentration. So what it means by first order, this is the rate that is equal to the maximum velocity and is independent of the substrate concentration. So we use this to measure substrates. But when the substrate concentration is higher than the Michaelis concentration, the rate of reaction is zero order with respect to the substrate concentration. So zero order, what it means is a chemical reaction in which the rate of reaction does not change when the concentration of the reactants grow or decrease. Okay, so the rate of this reaction is always equal to the rate constant of the specific reactions, since the rate is proportional to the zero power of the concentration of the reactant, and first order with respect to the concentration of enzyme. So we use this in order to measure the activity of the enzyme. So there are many factors that can affect enzyme reaction rate. One is through environmental factors such as the presence of inhibitors. There are two different types of inhibitors. It can either be irreversible. So irreversible inhibitors form a very stable interaction with the enzyme. They are covalently bound to the enzyme. So this renders this enzyme to be useless. It can no longer function, okay, and hydrolyze the substrate of interest. Okay, example of these types of irreversible inhibitors will be cyanide. Okay, and we also have these reversible inhibitors that can form this non-covalent bond with the enzymes. So with these reversible inhibitors, it can either be competitive inhibitors, where the substrate and this inhibitor will compete for the same active site, okay, the active binding site of the enzyme. Right, so with this, let me just draw here. Okay, so if you look back at this, okay, the graph of this enzyme kinetics, okay. So let's say for the competitive inhibitors, you have right? This is the Vmax, right? So with this, if it's a competitive inhibitors, what is going to affect is going to increase the Michaelis constant value, but it's not going to affect the maximum velocity of the enzyme, right? So this is without inhibitors, but with inhibitors is going to, has to be something like this. Okay, it will increase the Michaelis constant. For the non-competitive inhibitors, the inhibitor is not going to compete with substrate to the same active site, but it will bind to a distinct site. Okay, so once it binds, it will cause the enzyme not to be active. This can either be pure or mixed. Okay, so for non-competitive inhibitors, it will be slightly different because it will not change the Michaelis constant, but it will reduce the maximum velocity of the enzyme. Temperature can also affect the enzyme reaction rate. Enzyme, we know they are most active at their optimum temperature. Okay, if it's at lower temperature, they will show little activity and they will lose most of the activity at high temperature due to the denaturation of the enzyme itself, as I mentioned, because they are protein. pH can also affect enzyme reaction rate, as enzymes are most active at their optimum pH. So enzymes that contain functional group of amino acids with proper charges at optimum pH. Okay, so they will lose activity at lower or higher pH as the secondary, tertiary, and possibly quaternary structure are disrupted. So there are different classes of enzymes. Okay, they can be further classified into either they are oxidoreductases, where they are involved in the redox reaction. Okay, the enzyme can also be transferases, where they transfer groups of atoms, hydrolysis, where they hydrolysis compounds into smaller product. Okay, or they can be also lyases, where they add atoms or they remove atoms from a double bond. Okay, enzyme can also be isomerases, where they rearrange atoms. For example, they isomerize L amino acids into D amino acids, or they can also be ligases. Okay, so this where they use ATP and energy to combine molecules. In food system, however, I'm just going to further elaborate on two of the enzymes that are known to play important role in the food systems. One is hydrolysis and the other one is oxidoreductases. So you're going to see the beneficial property of enzymes, okay, the enzymatic reactions for in food, as well as what are the negative effects of this enzyme, okay, in the food application. Hydrolysis, these are enzymes that catalyze hydrolytic reactions, okay, where example for hydrolysis is lipases, esterases, aminases, peptidases, and proteases. Here, just to give you an example of starch, okay, that can be hydrolyzed by the action of amylases. So there are different types of amylases. It can be beta amylases, alpha amylase, pulilanase, and glucoamylase. So each of these amylase enzymes, they have specific target site where they are going to hydrolyze. They can either be exoprotease, okay, where they digest at the end of the starch, or they can be endoenzyme. Okay, we target the middle of the structure of this starch. Okay, and they either enzyme that hydrolyze the amylose or the amylopectin structure. So amylases is important, especially for the starch liquefaction, and they can also be used as anti-stalling agent. One example in Malaysia is that for the making of dodol. There's one, Mardi has developed a methods of method to prolong the shelf life and as well as the maintaining the quality of dodol so that they didn't become hardened, okay, by adding amylase to the dodol. This helped dodol to be soft and stored at room temperature over time compared to if you didn't add amylase. Another example of hydrolysis enzyme will be pectinase. These are the enzyme that is used as gelling agent as well as to thickening or to water bind as well as stabilizing in food. Example given here for pectin methylesterases. These are the enzyme of pectin that help in demethylate polygalacturonide and leads to the formation of pectic acid. So pectic acid help in stabilizing juice. Okay, it prevent coagulation of proteins in juice. Okay, so they remain homogeneous. Okay, and we also have here polygalacturonase. Okay, that can cause delay of the polygalacturonide by the endo PG or by exo PG. So this help in depolymerize pectin and lead to the production of digalacturonates. Okay, digalacturonates. Okay, this can be used as a clarifying agent in juice, and as well as pectinase that help in catalyzing trans-eliminative cleavage of pectin into oligogalacturonides. Okay, this can also be as a clarifying agent in juice. We also have other different types of hydrolysis, such as lactase, that can convert lactose to glucose and galactose. Okay, breakdown of this disaccharide into these reducing sugars. Okay, sucrase or invertase can also further convert sucrose to glucose and fructose. That help in improving texture, flavor, and bioactivity of the food. Okay, example given here will be meat. Okay, enzyme can be used to tenderize meat. Okay, example of meat tenderizer enzyme include plant proteases such as bromelain and papain from pineapples. Enzymes that belong to the oxidoreductase category. Okay, this is just to show you another example where this enzymatic reaction is unfavorable. So these oxidoreductases, they can catalyze the transfer of hydrogen or oxygen atoms or even electron from one substrate to another. Since these are redox reaction, the electron donor or acceptor is also required to complete the reaction. Okay, so one application of oxidoreductases is the polyphenol oxidase. This is oxygen transferring enzyme that are involved in the enzymatic browning. So this can be observed in cut fruit, as well as in shrimp. Okay, shrimp can also undergo this enzymatic browning. So this reaction is unfavorable because it reduces the shelf life of the fruit and reduce the sensory acceptance of this fruit as well. Okay, so what happened in this enzymatic reaction, enzymatic browning, is that you have monophenol, which is colorless in your food. Okay, and then this monophenolase activity will further catalyze the reaction to form this orthodiphenol, which is still colorless, but then you have this diphenolase activity, redox reactions where this then will transform into orthoquinone, which is the browning pigment. Okay, in food industry, there are many methods of how you can inhibit these polyphenol oxidases by adding different inhibitors. Can be acidulant, for example. By lowering pH will help in denaturing these enzymes, or by adding this chelating agent. Okay, again, this will inhibit the activity of this polyphenol oxidase, or through competitive inhibition or the addition of antioxidant will prevent the formation of this conversion of this monophenol into orthodiphenol, as well as for the orthoquinone. Okay, so that is just to wrap up on the enzymatic reactions. So the application of enzyme in food system and what's their importance. Of course, this is very brief and it's just an overview. To read more and to learn more about these enzymes, Okay, in food chemistry, I would advise you to read chapter 6 in the Phenema food chemistry textbooks.